Hi guys, I'm Claire, and today I'm going to be telling you guys about gel electrophoresis, what it's used for, and how it's done. So, gel electrophoresis is a common lab technique used to separate DNA fragments according to their size, and it heavily relies on an electrical current, and it is used to compare different samples with different lengths of DNA fragments. It's very common. There are numerous applications in the lab and the real world, which we'll be going into a little bit later. And there are also many different forms of gel electrophoresis. Okay, so this is the general setup of gel electrophoresis. So you have the agarose gel. You have the gel electrophoresis box, which is usually clear, made of plastic. You have your DNA fragments, which is what you're actually going to be analyzing. And you also need a reference sample with known lengths of fragments so you can actually kind of compare to see how long your fragments are. You need electrodes, power source, and wires for the electrical part. You need a buffer, which is usually a liquid solution or all the time. And you also need ethidium bromide and a viewing device to actually be able to see the gel electrophoresis that you did at the end. So preparing agarose gel is the first step to gel electrophoresis. So agarose is a helical matrix comprising of many pores for molecules to pass through, and smaller molecules will pass through the agarose better. And it can be made by dissolving an agarose tablet and heating it. And heating it to obtain the liquid form is essential at first because you need to put it in some sort of mold with a comb, as you see here in the image, so that you can get your desired shape of hardened gel for the actual electrophoresis. So the next step you need to do is load your samples. So all of your DNA samples should be placed in ideal conditions before doing the electrophoresis and transferred using micropipettes because you need very small amounts. And the hardened gel that you should have made should have little wells or like holes that are pretty small, which is why you need a micropipette. And usually your samples will have some sort of dye or color so that you can see where you're putting in your samples and make sure that it's not leaking out or that you put it in wrong or that you broke the gel somehow. And you place the samples on the cathode end, which is the negative end. So if you have the electrophoresis here, you're going to be putting in the samples here in the negative end, and they're going to be traveling to the positive end. Okay, so you need the buffer solution next. You're going to pour it in the gel electrophoresis box when the well is already there. So a buffer, the definition of a buffer is a solution that acts to balance the pH um, and keep it pretty neutral. So what the buffer does is it has ions, which are charged atoms, and that kind of carries the current and helps the gel electrophoresis proceed. And it also helps to maintain the pH as well. But the choice of buffer will vary depending on which type of substance you want to test. Okay, so now we have some quick quiz questions to test your understanding. So what is the substance which is used in a gel? If you want to think a bit on any of these, you can just pause the video. The answer is agarose. Next question, which ends are the samples micropipetted into? The answer is the cathode end, which is also the negatively charged end. Okay, so now to actually run the gel, you need to activate the electric current with the wires and the power supply. So it runs through the buffer, as I said before, because it has those ions, and the electric current moves towards the positive end, and the fragments will also move with it because DNA is negatively charged. So it's attracted to the positive end because negative and positive attract each other, as you may know, and that's how the fragments kind of proceed down. And the runtime for gel electrophoresis is about 60 to 90 minutes. And The shorter bands, as we mentioned before, shorter bands will go through the pores of the acros better. Um, so you see the shorter bands in this image in the bottom right will move down farther than the longer bands. And that's kind of how you can analyze your samples. So after you run your electrophoresis for 60 to 90 minutes, you can read the gel using your UV or your device. It lights up and you can place it on. So each sample is separated into component macromolecules based on their size, as I mentioned before, and the ethidium bromide or another staining solution is placed so that you can actually see your measurements and observe it and record it and determine the length if you want to, or compare it to another sample. And so now you need to record the bands. So each sample's fragments travel vertically down, and you have your bands if you're actually reading the electrophoresis, and they're pretty thick, so you can see them with a naked eye. And this is where you compare your sample that you already know, the reference sample, and that's kind of your reference for approximating the length of your DNA pieces. So the analytical method for actually reading these is plotted through a standard curve, which is based on a log scale because these numbers get pretty big. And known standard curve plots of similarly length fragments can be used to approximate unknowns, because you had your reference sample, which you know the length of. And the graph is usually base pair versus the distance of the fragments traveled. So what are the actual applications of gel electrophoresis and when will we use this? So as we mentioned before, it separates DNA fragments and you can use that for fingerprinting in forensics. You can analyze the results of a PCR, which is basically a method to make many copies of DNA. You can identify unknown restriction enzymes based on their cleavage patterns. You can analyze the genes of specific illnesses, and you can profile DNA to identify different species, or you can even do it to humans. As I mentioned before, it's very useful in forensics. And yeah, the last slide or the last point, attribution of genetic samples to records. So it's really useful in any sort of genetic testing or biology. Yeah, it has many applications. So we have a final set of questions to test your understanding. What type of analytical plot is used to graph results? And you can kind of see the answer on the graph, but the answer is a standard curve and it's based on a log scale. This is a harder question, so it's double points. What is the solution used to view the fragments of the gel when you're done with electrophoresis? The answer is ethidium bromide. And that's it. Hope you learned a lot about gel electrophoresis. If you have any questions, you can email us with the link or the email in the description. And hope you learned a lot. Have a good day. Bye.